Okay. So, as most of you, I think, know, it's been observed that spontaneous fluctuations in bold-weighted MRI time series data are highly correlated between regions with related brain function. And a very important aspect of this observation is that there's a high degree of specificity to this, and that the correlations that are observed are highly correlated between regions that are functionally connected, as shown here in the motor cortices, but regions that aren't functionally coordinated do not show this, this degree of similarity. So this makes this actually useful as a way to look at functional networks in the brain. Now, traditionally, the analysis strategies that were employed when this was originally observed consisted basically of just doing bandpass filtering and a temporal cross correlation. Now, this is referred to as a seed-based analysis technique, where you define a specific seed region and then just look at all the highest, most highly correlated regions across the brain, as shown here in the auditory, bilateral auditory cortex. So I'm trying to stay away from multiple clicks here when there's no. There we go. Okay. So what I've been asked to do here is assume that the people in the audience, the people attending this, are already familiar with cross-correlation type of seed-based analysis, and restrict myself to talking about the more complex network analysis techniques that have become somewhat common in resting state data analysis. And just for my own curiosity, could I see a show of hands, people that are familiar with doing seed-based correlation analyses in resting state data? Okay, so most of the people here have done this. Okay, well, I just briefly want to go over the pre-processing steps that are necessary for resting state data, because it actually is advantageous to do some things that are different than task-based fMRI. Most bold effects are limited to below 0.1 hertz, so you can enhance the signal-to-noise ratio of your effect by doing a low-pass filtering. Where you do this isn't really critical. Now, for slower volumetric acquisitions, at least traditional type of acquisitions, the specificity can actually be dramatically improved in a lot of cases by correcting for physiologic noise, which can be done in a number of ways. And I should point out that the newer multiband techniques, which we're going to hear about later on in this session, allow much faster acquisitions that directly sample these, that don't require these retrospective techniques. The 0.1 hertz digital filtering takes care of that. Slice timing correction in the low-pass filter data is not critical, but out of principle, it's nice to do it. Motion correction, of course, is always a nice thing to do. And it's been shown, it's very important to point out that it's been shown that there is a global signal that's present in most of the gray matter in the brain. And it's necessary, if you're looking at single subject results, it's not really necessary to worry about this too much. But if you're going to combine data and want to establish accurate p-values, it's necessary to correct for the global signal. And this is normally done by using partial correlation, but there are other ways to do this as well that are in the literature. And another thing to point out, which is true for all bold techniques, is that there are some patient populations out there that have compromised vasculature and will actually have a different vascular response given a certain amount of neuronal activation. And there are ways that have been presented to deal with that as well. There have been several processing pipelines that have been published in the literature over the years. Most recently, and easily available, is the HCP processing pipeline, which one can look at here. Now, I'm going to pattern my talk after a review paper that we published a couple of years ago. A group of us published in the Journal of International Neuropsychological Society. I'm going to cover briefly structural equation modeling, which is a hypothesis-driven method using a a priori established model. Independent components analysis, which has perhaps been around the longest of complex network analysis methods, and it's just been shown to be very robust. I'm going to cover a few common spatiotemporal clustering methods that are somewhat common. I'm going to spend quite a bit of time on graph theoretic methods, because this is actually becoming a very popular method to look at sort of global connectivity in data. And then I'm going to just spend briefly some time on dynamic functional connectivity. Now, structural equation modeling, this is a method that allows you to test an a priori defined model of the pathwise dependence of observable correlations or covariances in a set of data. Now, the application to fMRI, now, so this, as many of these complex network techniques, they're borrowed from the social sciences. So structural equation modeling was originally developed to look at issues that are present in social science. It is possible to apply this to fMRI or connectivity studies in order to test models of functional and or neuronal connectivity. And I'm going to show a brief example here that I've shown a number of times over the years that illustrates this method. But I should point out that there's a very recent nice review by Gardia Olmos in Frontiers of Behavioral Neuroscience that I encourage you to look at if you're interested in this technique. The basic idea is that you specify a path model of observables that you have in your data. And it's very important that the model that you establish can actually be tested with the data that you have. Okay, this is called the identification status of the model. And then once you have that, you can use a covariance matrix between your observables, or in the case of resting state fMRI, a correlation matrix. You can estimate the model parameters via regression and determine a goodness of fit to your model. Now, there are a few software packages that are available out there to do this. The common ones are LizRel, AMOS, and EQS. An example. So I show here an example where you can actually produce a model that will test the connectivity using structural equation modeling of subcortical and cortical motor cortex gray matter structures. And so what I do here is I use the model that was proposed many years ago by Grafton and DeLong, connecting subcortical thalamic and basal ganglia motor elements to the cortical motor elements with basically path directionalities. And using this, we can define a path model based on the observables that we can get in a resting state data set. Now, we know that if we do a complex finger tapping task, we actually get these regions of the brain that are active. And if we look at the model, we see that we can actually pretty easily identify these 5 regions of this cortical subcortical motor network. Okay? So consequently, we can actually construct from Grafton and DeLong's more complicated model a more simple model that we can use to test our resting state functional connectivity data. So what we do is we just define the ROIs, do the temporal filtering, and then we just form a correlation matrix for each of the regions. And using that correlation matrix, we can submit it to, I used LizRel here. And what we get are, in these 2 cases, where in one case we had the same subject at rest, and then we had the same subject performing a continuous finger tapping task. And we submitted both of the correlation matrices to LizRel, and what we got was, in both cases, the model fit is good, with good confidence levels. And what we see are the path coefficients for each of the connections. And the interesting thing to note, which actually makes a lot of sense, is that when switching from rest to tapping, what you see is that there's actually an increase in the coupling between the thalamus and the cortical motor elements. So this is something that structural equation modeling would be good for testing, which testing between 2 states or comparing model adequacy or connection strengths between populations. And this has been done in some in the literature, and I encourage you to look at the review if you're interested in this. Now, as I said, perhaps the complex network strategy that's been around longest, mainly because it's just been shown to be so robust in its applicability to many different data sets in many different situations, and that's independent component analysis. Okay, and so the idea behind independent component analysis is that it assumes that your time series data are related by simple linear transformation to a set of spatially independent components. Now, I want to point out here that what I'm going to describe here is spatial ICA. And this is because for a long time, due to the temporal degrees of freedom, basically spatial ICA was computationally more accessible. But now with new multiband techniques where you actually can have a lot more temporal degrees of freedom, temporal ICA methods are now becoming more common. So you can simply just change spatial for temporal and everything that I'm saying here, and the idea is the same. So independent component analysis will separate spatially independent sources that contribute to the entropy of the system. Okay, and this was originally proposed by Bell and Sejnowski. And this is a computationally intensive methodology where PCA techniques are a lot of times used up front to reduce the number of degrees of freedom. Now, some caveats to independent components analysis that are important to recognize as people using this technique is that you actually are required to define a priori the number of independent sources in your data. Now, a lot of techniques have been developed recently that actually use data-driven methods to try and estimate the number of sources that are in your data, but it's important to understand that your results will critically depend on this parameter that you put in to the algorithms and separate the sources. It's particularly important when doing group analyses. And the other thing is that it requires a post hoc interpretation of the results, of the spatially resolved components. You're reduced to, at the end, identifying what you consider to be functionally interesting components that are a result. Mathematically, as I said, your resting state data set Y here is defined as a linear combination of what's called a mixing matrix and your basis set of spatial maps. And the idea is that you find this mixing matrix such that this function of your spatial maps is minimized. And as an example, the typical forms of F that are used are mutual information, which is what's called the Infomax algorithm, or neg entropy, which is a form of decreasing entropy, which is how the fast ICA algorithm works. And I also like to point out that PCA is a special case of independent components analysis where F here is actually the covariance of the data. It's a simple example. I have six source images here that we've mixed. Okay, and to show that PCA is not appropriate for this, I show what you get with a PCA to try and deconvolve this mixture according to variances in the data, and you see you get something that doesn't make any sense. However, ICA does actually quite a good job, and to be clear, the components that are resolved with ICA, and here we knew that there were six sources, so we put in that there were six sources, and we got a pretty good job of combining the original data to within polarity anyway, in order. And so here's an example of the standard resting state networks, as they've come to be called, that you get with a typical independent components analysis. And this is what you get from performing this data, this technique in almost anybody. Okay, and that's what's so nice about the technique, is that it's very robust. And then group analyses have been developed, typical software that you can get freely to do independent components analysis. I think the first one that was available was EEG Lab, and then for a long time now you've been able to use Melodic from FSL, and Vince Calhoun's group has produced GIF that actually specializes in allowing you to do group ICA techniques. Now moving on to spatiotemporal clustering methods. It turns out that the connectivity maps that you see, even from a seed-based correlation network analysis, are essentially mapping out regions of spatiotemporal clustering. As an example of this, and if those of you that aren't familiar with the Instacore from the AFNI toolbox, I highly encourage you to have a look at it. What it does is it allows you to surf spatiotemporal correlations in your data. If you see the cursor here, this is running through this one slice of the data and showing a correlation map to the rest of the brain. This slice happens to be basically in the same slice with the area of the posterior cingulate that's involved in the default mode. And as you go around, you see the shifts in the spatiotemporal correlations that indicates changes in connectivity of the seed region. And when you hit the posterior cingulate, boom, you see the default mode network light up. Advantages of using clustering techniques is that they're model-free. They make no assumptions about the temporal distribution. However, you are required to parcel the voxels and downsample the spatial resolution. A few of the more common methods, there's hierarchical clustering, which is actually the first technique that was employed in resting state data. I'll talk about in a minute. Using this, you can form clusters according to some hierarchical models, either going from the bottom up, where you have a large number of voxels and you combine them together to get a smaller number, or top-down techniques where you start with one big cluster and break them up according to some metric. It's necessary to define a metric, for instance, a distance between your parcels or voxels that define a group, and then you have to establish a linkage criteria that allows you to combine these voxels based on your metric. And then the technique then is just to either reduce or combine clusters, either up or down, based on the direction that you're going, in order to define what your cluster structure is. K-means clusters is a different technique where you actually a priori define, either randomly or with some other method, K initial clusters. That's the name K-means algorithms. And then you iteratively assign each voxel to the cluster. Once you have these randomly assigned initial K clusters, you can then iteratively update the mean or centroid after each assignment step. And when you iterate this, essentially when there's no change in the mean or no significant change in the mean or centroid, you're done with your reassignment and your K-means clustering is done. Spectral clustering is another spatiotemporal clustering technique that's done. It's similar to graph theoretic methods that I'll describe in a minute. Martin van den Heuvel is the only one that I'm aware of that's actually published a paper that uses spectral analysis. It's called a graph cut method. As an example of a hierarchical clustering, what I show here is basically just a bunch of data points in two-dimensional space. And we define what's called a dendrogram based on the Euclidean distance, where you basically just successively, so this is a bottom-up approach, okay? So I define the Euclidean distance as our metric, and so all I do is hierarchically create the dendrogram where the closest distance is these 2 points, so that forms one cluster, and then the next closest 2 data points are these two. That's already a part of this cluster, so we cluster this with this point, and they would basically just move up to the next set of clusters, okay? And so once you get up to the point you only have one cluster, but you actually have a scale here where this is the distance at this level of the mean distance of the data points in your data within each cluster, you can define a cut. And so if I define the cut as I want the mean distance within each cluster to be 1.2 millimeters, centimeters. No, this is millimeters. So if I want it to be 1.2, what I end up getting, I set a line here, and I can see that I get 5 clusters. Okay, I get this big cluster here, okay, this one here, and then I get this small cluster here. I get these two points as individual clusters, and then this other one. So that's basically how you do hierarchical clustering, and there are more sophisticated techniques to do this, to establish this cut technique. As an example here, I show a result from Shams et al. Stephen Strother's group, where they actually used a top-down hierarchical clustering technique, where they constructed their dendrogram based on the homogeneity of the voxels, of the temporal behavior of the voxels within the cluster. And they just iteratively broke up the least homogeneous network from the others until they got a structure that looked like this, which you can see does actually represent what we consider to be sort of basic functional units of the brain. Graph theoretic methods. Okay. So graph theoretic methods, basically what this is doing is actually using an entire sort of theoretical body of techniques that have been established to analyze what are called graphs. And anytime you can actually take something that you can establish as a set of nodes, and if you can take your data and actually establish it as a set of nodes, you can make a graph. Because what the graph is composed of your end nodes that you've established and edges, which are defined as the relationship between each of the nodes. Okay? So once you have a group of nodes, you can calculate what's called the adjacency matrix, which is the weight or the metric of the relationship between all of the nodes. All right? Now I want to point out that when applying these techniques, graph theoretic techniques and some of the clustering techniques that I mentioned earlier, it's necessary, because of the computational effort involved, it's necessary to downsample the spatial resolution of your data. Typically for a 3T data set, you have something like 100,000 voxels in gray matter, and it's 7T data in ultra-high fields, you can have 500,000 tissue voxels, and this is basically not doable for these techniques. There have been a lot of techniques that have been proposed to do this parcellation, and I was very happy to see that the organizers of this arranged for the talk that's coming after me to be dedicated to a parcellation technique. So I'm not going to spend a lot of time on this, simply to say that it is necessary for these techniques to have a reliable method of parcellation that reduces your data into parcels. And each of these, and it's important to say that each of these parcels is assumed to be represented by a single time series. So all the voxels in these are represented by a single time series. Interestingly, there was a recent special issue of NeuroImage on segmenting the human brain, And in there, there's a really nice review that compared a bunch of commonly used parcellation techniques. Sadly, and this is a cautionary note, it's interesting, it is very important to understand the parcellation method that you use, because there's really none of these have been determined to be optimal for all of the things that you might want them to be adequate for. As I said, you start with nodes and edges. These edges can be either binary, that is, there's a connection 0, there's no connection 0, or there is a connection. More typically, you use the temporal correlation between the nodes in a resting state analysis. And most of what I'm talking to came from a review that was published in Neuroimaging Clinics North America by Medallia. And I encourage you to look at this. So here's an example of an adjacency matrix. Okay, right hemisphere, left hemisphere, cerebellum. Now, there are 3 domains typically that graph theory limits itself to, breaks down into. There's global metrics, there are intermediate metrics, which look at intermediate scale, and there's looking really down at the local domain. Example of local network analysis metrics. Now, in the local network analysis, we're looking at properties of an individual node. Okay, there's degree, for instance, which is the amount of connectedness of that node to the rest of the network. There's clustering, basically how much an individual node clusters with its neighbors. There's betweenness centrality, which basically is a measure of how much a node is involved in short paths across the network. Intermediate network analysis looks at the arrangement of nodes into communities, okay, characterizes varying degrees and qualities of modular structure, and the metrics that here characterize the extent to which nodes exhibit non-trivial community organization. Some examples: core periphery, which basically defines how many primary nodules are connected to sparsely connected nodes. Modularity, there are few modules with intervening nodes. And then this has actually become kind of important and very popular, which is the concept of a rich club, which says that basically nodes that actually are connected to a lot of other nodes are also connected to each other. Global network analysis. The nice thing about these is that they actually give single scalar metrics that represent your entire network. Simplest might be density, which is just the number of existing edges compared to possible edges in a binary classification. Lattice would be, there's a high degree of clustering but long path lengths. Randomness of your network would be a lack of distinct modular organization and short path lengths. And I'm on red. Small world metrics, which is clustering with short paths and long distance connections. As an example, I want to show an example of a graph theory analysis that was done where the Cleveland Clinic participated in a Huntington's disease. It's a data set of Huntington's disease subjects where there were 4 groups that were defined. The nice thing about Huntington's disease is that it's actually has a genetic identifier. And so there was a group of controls that were gene negative. And then in Huntington's disease, based on the degree that the genetic defect is present, you can define the risk of conversion from prodromal to clinical Huntington's disease as low, medium, and high. And so these groups were broken into low, medium, and high. And they were parceled into 300 nodes. We used the brain connectivity. There are a number of toolboxes that can be used for graph theory. This is a popular one because it was developed specifically for resting state techniques. We evaluated global and intermediate and local network measures. So interesting result. This is one of the figures from this where we actually show the groups where the degree of the node. So these are regions where the degree of a node had a significant effective group, and we show in each of the nodes the breakdown across all 4 of the groups. A much more interesting, I really like this plot, okay, because this is basically the rich club ratio, okay, which is a metric that comes out of the graph theory analysis with regard to the rich club, which I described a second ago. And what you see is that the gene negative groups and low, medium, and high all break out in a very specific and definite way according to their risk of conversion to Huntington's disease. So what this says is that the network organization of the brain in Huntington's disease basically is affected by the proximity or risk of conversion. Okay, so I'm out of time here. I had a couple of slides that I wanted to show basically on dynamic functional connectivity. This is a concept that, and I'll just show one of the movies here. So what this is a sliding window of 90 seconds showing, supposed to be showing, correlation to the posterior cingulate. So this is let me see if this one will run. Oh, the problem is it's not looping. Okay. Can I run it again? Yeah. So this is a 7 Tesla data set, and this is just a grayscale correlation matrix of the correlation to the posterior cingulate. And what you see is there's a shifting pattern of correlations. And so this is what's called a sliding window analysis, which is used, which is one way that people do functional connectivity analysis. Another common method is which is actually becoming more prevalent, which is time frequency analysis, which uses wavelet transforms to break basically a set of time series data up into basically time frequency planes where every voxel is represented by a wavelet coefficient. And then you can actually do statistics on these time frequency planes. I didn't really want to say any more on that because it's really kind of early on in the dynamic frequency connectivity. There really are no well-established metrics or statistical methods. But I do recommend, actually, I do recommend, do I have it here? There's a review, yeah, here. So this review just came out. It's Katie Chang, Mika Ribanoff, and Yingwen Chen. They have a nice review of pros and cons of dynamic functional connectivity analysis. So in summary, structural equation modeling requires an a priori structural model. Something I didn't mention. It allows latent variable modeling, so non-observables can be included in your model. I didn't point out, but I should say that this is actually not suited for comparing different models on a data set, but rather comparisons are really only valid when you have a good model. Independent components analysis, well established. Detailed results depend on knowing the underlying sources. Clustering methods are model-free, computationally intense, somewhat difficult to validate. Graph theoretic methods, there's an extensive body of theory and metrics behind this. This technique requires parcellation of volumetric data. And as I just said, dynamic connectivity, which is kind of new on the scene. It's kind of exciting and very interesting. So I'll stop there. I apologize for running over.